I'm just going to give a little brief outline of the presentation today. And I'm going to be talking about some work from our group here at Northwestern. And I'm going to be talking about two different community-based studies that we've been doing where we looked at light exposure. And then I'm going to talk about some experimental studies that we've done in the laboratory that sort of complement those community-based studies. So this slide must look familiar from Dr. Dibner's introduction. And the work that we do in the group here at Northwestern, we're very interested in the effects of light and how it influences the circadian system, but also how external time cues impact the circadian system and sleep-wake timing. And so today I'm going to be talking about light, but we also do work looking at physical activity, food intake, melatonin, and body temperature. And as Dr. Dibner mentioned, the circadian system affects a lot of peripheral tissues and clocks throughout the body. And so the focus of my talk today is really on metabolism and the effects of light on metabolism and the work that we've been doing in that area. So a lot of the work that we do when we measure light and when we measure rest activity cycles in humans, we use these rest activity monitors. And this is just an example of a rest activity monitor recording for about 7 days from a shift worker. And each line here is a day from noon to noon. So 1, 2, 3, 4, 5, 6, 7 days. And then the dark bars here are activity, and the yellow bar here is light level. And the centerpiece here is midnight. So in the shift worker, they're awake at night and sleeping very, very short periods during the day when we have no activity here. And then after a couple of days of doing night shifts, they sleep during the day, they work a few more night shifts, sleep at night, they're awake at night, and then sleep again at night. So their schedule is very perturbed. And they're also getting light-dark schedules also are perturbed. We also study people with delayed sleep-wake phase disorder, for example, and those are people who tend to be night owls, and they have difficulty falling asleep early as they would like, and they tend to sleep very late. And so in this case, these people are getting a lot of light at nighttime, and then sleeping all the way through till noon, sometimes 1 or 2 in the afternoon. We also look at people with NUN24 disorder, and in this case, we have somebody who is awake at night and sleeping during the day for about a week, and then their clock kind of free runs a little and drifts a little bit later every day. And so they're getting light and dark at various times of the day. And then lastly, what we would consider a conventional sleep-wake schedule, where somebody is sleeping from about 10 o'clock to sort of 6 o'clock, a little bit longer on the weekends, and getting light during the daytime and less light during the nighttime. So that's kind of the sort of premise of some of the data that I'll show you from these community studies. And as Dr. Dibner mentioned, when we get light is very important. And so this is an example of a phase response curve to light. And when we get light in the evening, then we tend to phase delay the clock or shift it later. When we get light in the morning, we tend to shift the clock earlier or phase advance it. And just as an example of a phase delay, this is a single person's melatonin profile. Melatonin is a hormone that's produced at night, and we use that in human studies as a marker of the circadian clock. That in this case at baseline, we have a midpoint of sleep, midpoint of the melatonin at about 3 a.m. When we give a phase delaying pulse of light, we can shift that melatonin profile by an hour or so. And so the effects of light and when we get it are important, so that's something to kind of consider here. So what about these community-based studies? I'm going to talk about two different studies, one in pregnancy and one in older adults. And when we talk about light, it's hard to not talk about sleep. So you noticed when I was showing those activity records that the sleep-wake cycle or rest activity cycle very much impacts when we get light. And so there's a lot of literature out there that suggests that poor sleep, including sleep disorders, poor sleep quality, short sleep duration. We now know that the regularity of sleep is important, and also the timing of sleep are important. And so when we have poor sleep in all these kind of different facets of sleep, there can be changes in autonomic function, increases in inflammation, and in oxidative stress, and altered energy balance. And so this can lead to altered vascular and metabolic physiology, cardio-metabolic disease risk and or disease, and in the case of the study, one of the studies I'm going to talk about today, adverse pregnancy outcomes. And so what we were interested in doing, we know a little bit about the sleep, and I'll show you some of the data we have on sleep from that study, but we're also interested in looking at the effects of light and the light exposure in these people. So the study was the New Mum to Be study, and we had a grant to look at sleep duration and continuity during pregnancy. And these are some of my collaborators. So the study was of 10,000 women who, with their first pregnancies, who had single gestations across 8 sites across the US. In the sub-study where which we were involved in this sleep duration and continuity study, we studied about 900 women in the second study visit, which was about 16 to 21 weeks of gestation. We studied women who were 18 and who did not have pre-gestational diabetes or pre-gestational hypertension. And we were very interested in adverse pregnancy outcomes, including gestational diabetes, hypertensive disorders of pregnancy, preterm birth, and gestational weight gain. And we've published studies on all of those outcomes in relation to sleep, and then in some cases related to the light exposure. And so just very quickly, the women had 3 separate visits, one early in pregnancy, one in mid-pregnancy, one in later pregnancy. And in this middle one, we measured wrist activity monitoring, where we were able to record rest activity as well as light exposure. And so just to kind of give you a little idea of how these women slept, this is an example of the amount of time that women from various racial and ethnic groups spent in bed during pregnancy, in the middle of pregnancy. And then this is how much they slept. And to kind of put that in perspective, for adults, 7 to 8 hours is recommended, and that you can see that in some cases, these women are spending quite a long time in bed, but they're actually not getting very, very much sleep necessarily, so sleep efficiency is quite poor. So we showed in relation to metabolic function that late sleep timing, and in this case, late sleep timing was defined as a mid-sleep point that was after 5 a.m. in the morning, and the late sleep here is in purple. And this is women with gestational diabetes. And what we showed is that if you had late sleep, that you were more likely to have gestational diabetes than if you had earlier sleep time. And this was significant after adjusting for sort of age, ethnicity, employment, and insurance status. And the reason we control for these is that late sleep timing was significantly associated with being young, so 22 years or younger, being poor, and also being either a shift worker or being unemployed in this particular cohort. And this was the case both early in pregnancy and late in pregnancy, and that we also had similar results. This is with survey data. We also had similar results in the smaller sample where we had objective sleep. So why am I telling you about late sleep timing? Well, late sleep timing, when you're sleeping late, you're also potentially getting light at the wrong biological time. And so some of our follow-up studies are looking at just that. So the goals of this analysis were to characterize light exposure patterns during pregnancy across 24 hours and prior to bedtime, a time when light exposure is being shown to negatively impact sleep and metabolism, and across the waking day. And so we were examining gestational diabetes, but also I'll talk to you a little bit about a new study that's in preparation where we're looking at depressive symptoms in women as well during pregnancy. So the study where we looked at light in pregnancy and the risk of developing gestational diabetes was led by Minji Kim, a physician here at Northwestern. And in that study, we looked at light exposure, which is indicated here, as relative to time of sleep onset. So 0 here is when someone fell asleep, and then 24 hours before. And this is light exposure. And then this is activity exposure, activity levels at the same time. And what you can see is that in these sort of 3 hours before someone falls asleep, and even maybe 4 or 5 hours before someone falls asleep, that there's this difference in light levels between the women who had gestational diabetes and those that did not. Those who had gestational diabetes tended to have higher light levels prior to sleep. And so this is just a zoomed in version of that 3-hour window and 15-minute bins. And you can kind of see where the light levels are different between these two groups. And there is no difference in activity level. So it's not like they're more active and out doing things necessarily compared to the light levels. And what we found is that when you look at that light exposure 3 hours before bedtime, that it was associated with a higher risk of gestational diabetes. And what we did is we looked at light exposure above 10 lux. And the reason we chose 10 lux is there's recent recommendations that light exposure before bedtime should be less than 10 lux. And what we found was that women who were in dim light in those 3 hours before bedtime tended to have or be less likely to have gestational diabetes than women who were in moderate or bright light prior to bedtime. And this was significant even after adjusting for different confounders. We also showed that light exposure before bedtime, not surprisingly, was associated with sleep timing, sleep continuity, and sleep regularity, and it was not associated with sleep duration in this particular cohort. I think partially is because these women sleep quite a long time. So overall, objective and subjective late sleep timing is associated with gestational diabetes. Greater light exposure prior to bedtime is associated with gestational diabetes, and lower daytime light exposure during the is associated with depressive symptoms. And so sleep disturbance changes across pregnancy, and that sleep and light levels are potentially modifiable factors during pregnancy, a time when it's very important for both women and for the unborn children. So now switching gears a little bit to light at night. Light at nighttime can come from many different reasons. So you can have light on the room, we have a bedside light, we can have left the TV on, there can be light from outside, there can be light pollution. We can be sleeping during the day or sleeping late, so you're also sleeping a little bit during the day. You can turn the light on if you get up and go to the bathroom, or if you get on up to check something. And this is an example of a rest activity pattern where someone is getting up multiple times during the night and turning their lights on and getting light exposure in the middle of the night whilst, in theory, they're sleeping or during their sleep period. And so in this study of older adults, we were looking at older adults 63 to 84 years. It's about 1,400 people in this study. And we have a subset who wore a wrist activity monitor that had a light sensor. And so we were able to look at light exposure across the day and night. And in this case, we were interested in light at night. And we divided our group into those who had no light during the lowest 5 hours of light exposure across the 24 hours, and those that had any light during that time. And what we found in that study, and this is also led by Minji Kim, is that those who had light at nighttime, so I'm trying to find my marker here. Those who had light at night tended to have higher rates of obesity, diabetes, and hypertension than those who had no light during those lowest 5 hours of light at night. And that this was significantly, there was a significant increased risk of obesity, diabetes, and hypertension. And this is even after controlling for various things such as sleep timing and sleep duration, activity level, age, etc. So what about some experimental studies that kind of complement these sort of community-based studies where we show differences or relationships between light and cardiometabolic health? And so some of this work started when we were looking at light and activity in relation to BMI. And we did a study where we were able to create this metric called the mean light timing that we was in collaboration with Giovanni Santastasi, who's photographed here. And we took light exposure patterns across many days, across the 24 hours. This top panel here is just two examples. All the dots here are light exposure across different days, and the dark bar here is the average. This line here indicates 500 lux, which is quite bright room light. And so what we found in this study is that if you had a mean light timing, which is indicated by this little star here, that was earlier in the day, you tended to be have a lower BMI than if you had a mean light timing that was later in the day. So earlier light, lower BMI, later light, during the daytime, higher BMI. And so we were interested in why that might be the case. And so we did a study that was led by a graduate student at the time, Ivy Chung, where we gave light exposure, or blue-enriched light exposure in the morning and in the evening, and looked at its impact on metabolic function. And so this is just a little schematic of that experiment. We had two different groups. One group had dim light for a day, then the next day they were in dim light, except for a period of time in the morning when they were given bright light. And in the evening group, dim light for the first day, and then they were given bright light in the evening, and blood samples were taken during these key periods. And the light exposure was given with two of these blue enriched light panels, and in the room with also the room light on. And what we found is that when we shone light on people when they were eating, and even before they were eating, that there was these alterations in metabolic function. And this is indicating the HOMA-IR. And when we were in dim light, the HOMA-IR was low. In the blue enriched light, the HOMA-IR was significantly increased, both in the morning and in the evening. We also saw differences in insulin. And just to point out here, this is actually 30 minutes into the light exposure before someone has eaten that we see this effect. And then this data here is showing the effect after somebody has eaten, where insulin levels are higher in the blue enriched group than the dim light group, both in the morning and in the evening. We followed up this study more recently, and this is also led by Ivy and Dr. Daniela Grimaldi here at Northwestern, where we're now interested in looking at light exposure's effects on at nighttime during sleep on cardiometabolic function. And in this study, individuals either had the first night in darkness, or they were randomized to either a light condition or dim light. And then during wake, light was 240 lux, and at night it was dark or less than 3 lux, and when they were sleeping in the light condition, about 100 lux at nighttime. And what we found in this study, when they did an oral glucose tolerance test the next day, after sleeping either in the dim light or sleeping with the lights on, that there was alterations in insulin. So on day 2, in the room light condition, insulin levels were higher, and there was no difference in the dim light group the next day. So we also showed from this study that there was increases on day 2, or significant increases on day 2 in the room light condition in the HOMA-IR, which is a measure of insulin resistance. And we also showed that there was an increase in that early phase of the insulin response to the glucose challenge, and a reduction in whole body insulin sensitivity as measured by the Matsuda index the next day after sleeping with the light on. We thought some of these effects might be due to suppression of melatonin, but we did not see any significant difference between the room light condition or the dim light condition in melatonins. So the effect that we see is not necessarily related to melatonin suppression. But what we also did see is that during the night with sleeping the lights on, that heart rate was consistently higher across the night than when someone slept in the dark. And then someone slept in the dark, in the dim conditions, there was no difference at all. And interestingly, when we look at that early phase response, insulin response to the glucose challenge, that is related to changes in heart rate across the night, and in particular a change in the LF-HF ratio, which is indicating a shift from the sympathetic drive to dominance over vagal, and that we don't see an association in the dim light condition between these factors. So why does light matter? So light exposure during the day and night are both important for metabolism. I hope that the data I've shown you today, which is not all the data that out there, just some of the data that we have, that the general recommendation is that we should be getting more light during the day and less at night. And there's also some evidence to suggest that adequate light during the day can partially mitigate effects of light at night. Optimizing light exposure across 24 hours can be complicated to achieve. People have a lot of things going on, and there is a lot of external light at nighttime, sort of the sources of light at nighttime that we would need to reduce. And there's also things like individual variation in light sensitivity that we need to account for. We also don't know how much light at night is too much, and how do we balance the risk for falls. So how do we balance safety and avoiding light at night if you have to get up and go to the bathroom? You know, so you want to maybe have lights that are facing down, that maybe are you know, not very bright, and potentially even red or amber lights. And so I think one thing that we do know is that we still have a lot to learn about the effects of light on metabolism, and both during the day and the night. And I hope Dr. Harmson's talk will give us some greater insight into that as well. And I'd like to thank all of my collaborators and funding sources for supporting our work. Thank you.